Cool. Hi, everyone. I'm Michael. I'm a postdoc in the postdoc lab at the University de Montreal. And today I'm going to be talking a bit about how we can optimize spatial sampling to detect species range shifts. So first off, one of the most, it's my turn to fight with the clicking system, I guess. Yeah. Giant green one, it's not doing anything. The one above, oh, the giant green. Thank you, Isaac. So one of the most foundational questions we can ask about life on Earth is where different species live. And as we heard a bit in the first keynote on Tuesday, this is a question that we're actually not doing too great of a job of answering. But it's still a necessary thing to understand both what the composition of biodiversity is and how humans are changing it. So the way we typically do this is by using correlative species distribution models where we have a bunch of bioclimatic variables and georeferenced occurrence points, and we use that to predict where on Earth the suitable habitat for a given species is. But of course, as we know, species ranges are changing over time, both because of climate and land use change, and the result of this is that there are regions where species used to live that are being lost, here shown in red, and also regions that are being gained, shown in blue. So today I'm going to be talking about how we can use different algorithms for designing spatial samples in order to optimally detect where and when species ranges are changing. So just to take a step back, I think it's important to talk about when you're doing actual range prediction, there are multiple ways for a species range model to be wrong. They're not necessarily created equal. So here in the top right corner, you can see what's called a confusion matrix. So the columns there are what the true state of the world is, whether the species is present or absent at a given location. And then the rows are what our model actually predicts. So that diagonal, downward diagonal in blue, that's where we want to be, where the model is making correct predictions. But each of the off-diagonal entries represent different ways that the model is wrong. So for example, you could have a false positive where you're overestimating a species range, or a false negative where you're underestimating it. And the consequences of this could be different depending on what your objective in monitoring a range is. So for example, for an invasive species, underestimating the range could be more consequential than for like conservation of an endangered species, where you really want to avoid overestimating the range. So when you take this into the temporal context, we then have three different states. So you have the state where there's no range change at all, and then also range being gained and range being lost. So again, the downward diagonal there is where we want our model to be. This first example in blue is where our model is correctly detecting there's no change. And here what we're interested in today is these regions in green, where we're doing a good job of predicting where we're gaining range and losing range. But again, there's also multiple ways for the model to be wrong. So here in yellow is what I'm calling a missed range shift. I'm not sure if you can see that. It's a bit pale. But the idea is that this is places where the range is changing in the real world, but we're not actually able to detect it with our model. And so sort of the opposite of that is what I'm calling a hallucinated range shift. So this is where there is actually no range shift, but your model's telling you there is. And then finally, there's the last situation, which is kind of the worst case, where you're detecting a range shift, but you have it completely backwards. So this is really what we want to avoid. But my main point in showing these slides here is that for us to accurately detect a range shift, our model has to be right twice. So you have to get it right in the past and then right in the future. So here today, we're going to be focusing on optimizing for what I'm calling captured range shift. So this is times where the range is actually changing in the real world, and we want to maximize how much of that we capture. So that ratio of the green to that overall area of the two columns on the right. So how do we do this? Well, we're going to apply three different ways for designing spatial sampling. The first is random sampling. So this is the simplest. The idea is that every point in space has equal probability of being chosen. But this is not usually ideal for spatial sampling because, as you can see, there are a bunch of regions where the points are clumped, and then there are also places that are sort of systematically undercovered. And the idea is that if you have two locations very close in space, you're probably going to get very similar results from sampling there. So it would be better to spread that sampling out and to avoid redundancy. So another common method that is used a lot in the spatial sampling literature is called balanced sampling. So this is where sampling locations are very well spread across space, and this also has the theoretical benefit of the values of the predictor variables at your sampled sites tends to reflect the distribution across the entire extent, and that has a lot of really useful properties for inference. And then finally, we have a different method, which we heard about a little bit earlier today, which is adaptive sampling. So this differs from the other two because instead of choosing just a set of static sites to remain the same over time, you're actually using the model to update and inform where you sample, collect more data, and then you can sort of go through this iterative cycle where you improve your model and ideally do a better job than random sampling over time. So just a bit more detail on that. The method I'm using here is called uncertainty sampling. This is actually like the simplest form of active learning, which we heard a little bit about just before the break. And the idea here is that you're choosing to sample directly in proportion to the uncertainty of your model. So you select some sample size proportional to uncertainty, you collect more data, you fit the model again, you get a new prediction, you get a new uncertainty layer, you sample again, and in principle this improves how quickly you converge to the true distribution versus random sampling. So how exactly do we test these methods? So we're going to use a virtual species approach. So we're going to use the true bioclimatic variables for Quebec, but we're going to simulate virtual species niches by randomly simulating these different logistic response curves to each bioclimatic variable. And from that, you get a synthetic range out of that. And because we're using the real bioclimatic variables, we also have their projections into the future. So we can just project forward and pass it through the same simulated niche, and you get this simulated time series of a range shift. And for now, I'm just considering a case where the niche is static over time, just to keep things simple. That is a real valid concern to think about in the real world. But for now, we're just going to use the same niche across all the time points. And so what we're going to do is compare how each of these different sampling methods change as you increase the total sampling effort across all time points. And as a reminder, what we're measuring here on the right is the proportion of total area where the range is shifting that we adequately capture within the model. So we start with random samples, and you see you actually don't do too bad. Even with 100 samples across all time points, you get over half the captured range shift on average. And this starts to asymptote around 0.7. So that's pretty good, considering this is the simplest, most naive form of sampling. But we want to know, can we do better than this? So the next method is spatial balance. And we see something that's actually kind of counterintuitive, which is spatially balanced sampling does not improve at all. And this at first seems strange because there's a lot of literature and a whole lot of papers written about why spatial balance should improve your inference in a spatial context. But I think there's a reasonable explanation here, which is that when you have bioclimatic variables that are really highly autocorrelated, especially in a place like Quebec where there's a really strong north-south gradient governing the distribution of those variables, you can do a really good job of interpolating across space, even if you have some clumpy areas or some systematically undercovered areas. So what about adaptive sampling? Well, with that, you can see we do actually see a slight improvement around 5 to 10%. And this is good. This is showing that using the models to inform sampling does improve things, but it's not the whole story. And the reason is these are means across hundreds of different range shifts. And if you look at the variance there is a ton of variance around not just the adaptive sampling, but each of the models have, each of the sampling models have very similar variance in these contexts. So it's good, but we don't just want our sampling methods to do well on average. We want them to be pretty good for particular instances of species range shifts. So just very briefly, we can take a look at what's actually driving this effective predictability. And basically what this is saying here is that the larger the range shift is relative to the size of the range itself, the more, the better you're going to be able to capture the range shift. And this explains around 25% of the variance, which is not too bad. And so just to briefly conclude here, the whole idea is that you can test sampling strategies with simulation, and this is really important before we go and spend a bunch of money on a costly monitoring program to generate an expectation of how well we expect it to do. Adaptive sampling can do a better job than just straightforward static spatial sampling in improving species range detection, but there's still a lot of improvement to be done here for reducing the variability of these methods across different range shifts, because we don't just want this to be better on average. We want to be confident that we have a real narrow bound of how much range we expect to capture for a given sampling method and sampling effort. So with that, I have about a minute left, so hopefully there's time for questions, and thank you.
Thanks, Michael. And let's take one question, because you did that so fast. That was amazing. Michael, what's your name?
Super exciting stuff. And one thing I was wondering if you could modify this or are interested in. One of the consistent things we get with species distribution model is like, well, species have been, pushed out of the sort of like warmer parts of their ranges in some areas. And so could you use this sort of approach to say like, well, assuming there's certain types of biases, can they use that to help us decide, how bad our models are or how to improve them taking into account more of these types of biases?
Right, exactly. That's a great point. One of the methods that I think could really be used to improve this is using environmental variables like temperature to drive, in addition just to uncertainty. So there's a ton of information that could go into the, sort of that targeting of adaptive sampling locations that we're not using. But something like that where we know like the hot edge of a species range is likely to be lost much quicker than something in the middle, that's absolutely useful for this type of sampling guidance. So yeah, thank you.
Okay, and our second/first speaker is Millie Chapman. It's coming up in a minute. Contextualizing biodiversity data gaps in conservation decisions to inform targeted monitoring. Go ahead, Millie.
Yeah, this is right. Yeah, sorry everyone. My original title was a little bit ambitious as a first semester assistant professor, so you're gonna get what you're gonna get. Yeah, I'm gonna talk about some exploratory work, and yeah, hopefully a lot of conversation will come from this. Okay, so background I'm just gonna breeze through. Obviously, a heroic amount of effort is going into monitoring our biosphere and the ways in which it's changing, thanks largely to this community and the communities that stem off of this community. But despite all of this effort, of course, we have biases in these data. Not only biases that track macroeconomic patterns and accessibility metrics, but legacy effects of racist policies, armed conflict, colonialism are all reflected in when, where, and by whom our data are collected. And this is true on land, but it's also true in the ocean. One of my colleagues presented a paper we recently published on, yeah, just that these gaps are also really pronounced across the high seas, which have huge implications for decisions in that space. But I'm going to focus on land because that's what most of my work focuses on. Okay, so we've also heard a lot about what we use all of this biodiversity data for. Loads of different ecological insights. Models of species distributions, metrics of population change, identification of invasive species spread. The list goes on and on. And these disparities have implications, obviously, for these ecological insights. In a recent paper led by one of my colleagues, we show that the biases of biodiversity data towards low-income communities in the US, or low-income census tracts in the US and primarily white census tracts in the US, potentially misrepresent the environmental space of the United States writ large, which might be a mechanism by which these sort of biases could propagate through our ecological insights. Yeah, and we've started to explore some of these ideas. So here you can see on the x-axis mean household income of a census tract. This is just for the US, and density of GBIF observations. It's normalized by the mean number. Don't worry about it too much. The gist is that, unsurprisingly, we have less observations in low-income census tracts than we do in high-income census tracts in the US. And so the question is, does this propagate through our distribution models? So we take these observations, I guess you can see this down here, run some really simple SDMs, nothing fancy at all. I'm sure people are doing much more sophisticated work in this space in this room. And for 1,000-ish species, following another paper, this is not new techniques, and show that these patterns are kind of refracted into the SDMs. We can see sort of this low income bump in species richness projected into our into lower income, or less species richness in lower income census tracts than in high income census tracts. Of course, this could be a true ecological pattern. there could be less biodiversity or less species richness systematically in lower income census tracts. Absolutely possible. But when we look at like IUCN data and use like range map estimates of species richness, we don't get the same pattern, which again is not to say that those species richness patterns are right and the SDMs are wrong, but just to say that like there are potentially significant implications of these data biases on our downstream ecological insights. But what most of my work focuses on is not species distribution models or otherwise, but largely like how we use these ecological insights in decision-making processes. So it doesn't really stop at like learning about our ecosystems, but really these insights are informing pretty massive scale decisions and presumably going to impact the ways in which like billions of people interact with nature for decades to come. And so I'm going to focus on just like one of these 3 streams because I only have 5 minutes. Yeah, so species distribution models leading to conservation area planning. Don't really remember my logic here, but oh, okay, yeah. So we're often thinking about these ecological insights, and then maybe like as we've heard about a fair bit throughout the last session, like we can use sort of our capacity and our understanding of uncertainty in our distribution models or otherwise to potentially inform our data acquisition. That's great, but now we also have these downstream decisions, and we also have like a lot of human use of these data and of these models. So can we not only kind of stop at the ecological insights and feeding that into where we think about like targeting areas for data collection, but also think about maybe our conservation areas or a whole slew of decisions. There might be like typologies of decisions that these ecological insights are used for, and therefore like different implications of improving our ecological models. And yeah, can kind of that whole holistic scope of the use of these data, not only for ecology, but also for data, also for decisions and also for end users, feed back into our informed, or help inform our monitoring. Yeah, which I guess comes to this point about data in the decision space. So going back a little bit to GBIF data, when we look at kind of the distribution of the density of GBIF data mapped alongside the density, or like how much of area there is of different human footprints, we can see that like the GBIF data is like a little bit bimodally distributed, where we have a fair bit of data in like lower footprint areas, not like super low footprint areas, not super inaccessible areas, but like national parks, these sorts of things. I think this is just for the US actually. And then we of course have a lot of data in like cities. And we don't have so much in the middle, which is actually like probably where we're trying to make decisions, especially in the context of like strategic conservation. Like we're probably trying to, if we're actually trying to like expand area-based conservation, it's probably going to need to happen largely in this space that's of like middle human footprint. I'm gonna skip this bit because I'm running out of time. So maybe we can start to think about like these areas that we actually are thinking about expanding conservation alongside where there are data gaps, to think about like where we might actually have monitoring initiatives that could help inform conservation expansion. So if we look at biodiversity data gaps, these are just like the lowest 50% of data density pixels at 10-kilometer resolution. And if we look at kind of the same resolution of conservation priorities, I just pulled this from a global map that's been published. Maybe we could think about like which of those data gaps would be most helpful in informing, where we should invest, where we might invest in monitoring, because like we're probably not going to be able to invest in monitoring everywhere. So of course we can like look at overlaps of these maps and see like where there's gaps and where there's priorities and where there are both. But we can also think about this, of course, not as like a binary thing, because those are obviously like totally arbitrary thresholds, but rather look at like density versus conservation priority, and start to think about the ways in which we might prioritize monitoring towards these priority areas. Yeah, which I guess the key takeaway is just like, can we think about contextualizing these data gaps and constraining these data gaps to areas that are actually most relevant to the decision processes at hand? And of course, again, this is like a super oversimplified version of what is like a really complex problem, that there are a lot of downstream decisions, and therefore like using those decisions in the first place to inform monitoring could also be a very short-sighted way to go about monitoring. So this is something I'm quite excited to chat with people about more over beers later. And also with all of that, I think that there's like, of course, a really important discussion to be had around like how we think about human rights in the context of biodiversity monitoring and in the context of like proposing kind of massive scale monitoring of our ecosystems, that these monitoring technologies are ultimately rooted in kind of surveillance and military industrial complexes. And yeah, just thinking about like what community consent means when we're proposing really massive scale expansion of monitoring is critical. And with that, I don't have time for questions. Thanks.
Thanks, Millie. We have a lot of questions after. Okay, so we now have 2 speed talks. Do we have 2 speed talks? Oh, okay, there you are. The first one is Robert Lewis talking about Forest Web 3, mobilizing, harmonizing, and incentivizing forest biodiversity and environmental monitoring data through Web 3.0 technology. Go ahead, Robert.
Hello, hi, do you hear me? Okay, I'm going to present Forest Web 3. It's a Biodiversity funded project. It's a collaboration between NINA, which is the Norwegian Institute for Nature Research, which is where I work, and the University of Antwerp, the University of Bergen, and the Stockholm Environment Institute. And just to put this into context, we understand these alarming trajectories of biodiversity. But in order for us to understand for any given space or time where we fall on those trajectories, and also importantly to try to understand the impact and effectiveness of any restoration or conservation decisions, and or where we might need those most in space, we urgently need to continue mobilizing biodiversity data. Furthermore, if we want to bend the curves of biodiversity decline, we understand that we need systematic and sustainable behavioral change. So Forest Web 3 project is actually quite a simple one in the way it's organized. We have 2 main questions, if you like. How can we better mobilize biodiversity and environmental data? And then ask ourselves, how can we better utilize this environmental and biodiversity data? And the whole project is set within a behavioral psychological framework of NOZAC, which is trying to steward sustainable behavioral change. So the first objective is to ask, how can decentralized and distributed technologies help break down the barriers to mobilizing data? And so this harks into some of the pillars in the framework, the early pillars of how to make behavior normative, how to make it easy. And then the second objective is how can we then foster sustainable stewardship of forest biodiversity conservation through looking towards incentives like regenerative finance. And this harks then into the latter pillars of the behavioral psychological framework, which is how can we make behaviors rewarding. So previous work of ours have identified that distributed and decentralized ledgers, for example, blockchain technologies, founded in principles of trust, transparency, and control. And those are also the fundamental properties which individuals and institutions want when they come to mobilizing data. They want trust, they want transparency and provenance of the data, and they want authority to control. So we did a literature search to broaden our understanding of this, but now we're actually canvassing the wider academic community, and we're asking you to share with you your views of what the barriers to data sharing are and how certain technologies can help break down those barriers. So if you're willing to contribute to a survey of Forest Web 3, you can scan this QR code. And if you would like to understand a little bit more about how technically a decentralized and distributed data network exists, you can explore EchoChain, which was a previous project of ours, and a paper in Methods in Ecology and Evolution by Kjell-Erik Marstein, John Arvid-Gritnes, and myself from a couple of years ago. So how can then we incentivize more the mobilization of biodiversity data? Well, when we think about forests, globally, approximately 20% of forests are privately owned. And then when we zone in into particular regions like North and Central America, Oceania, and the European Union, 20% becomes more like 50%. Now, there's only really one bioeconomy for forest landowners, and that's resource extraction. And if we take the USA as an example, the majority of harvested timber came from privately owned forests. So private forest landowners can be really important actors to steward in and shift in or bend in the curves of biodiversity decline, provided that we can give them a choice between bioeconomies centered on resource extraction or bioeconomies centered on resource preservation. So as part of our work in NINA, we're exploring the voluntary biodiversity and carbon credit markets, and we're looking to see how they can be instrumental in giving landowners exactly this choice between resource extraction economies or pro-forestation economies. And where does Forest Web 3 come into this? Well, all of the mobilized data that we have in Objective 1, we're using it to improve the environmental and ecological realism of the models that underpin the crediting. But we're also trying to incentivize the forest landowners to steward in situ data that can help with the verification, so the measuring, the reporting, and the verification processes of these crediting companies to ensure that that is ecologically and environmentally representative, and also to ensure that this novel nature finance tech is indeed having a net positive contribution to the nature and the climate crisis. So if you would like to learn a little bit more, you can scan this QR code and it will take you to a poster just summarizing the project, which was presented a little bit earlier this year in Barcelona as part of a biodiversity project. But otherwise, thank you very much for listening, and if there's questions, I'm happy to take them.
All right, so unfortunately we're running a bit behind, but we're going to have a discussion period, so save your questions. So next up is Avery Hill, talking about uncertainty-driven sampling priorities, mapping where new iNaturalist observations matter most for California vascular plants.
Hello, everyone. My name is Avery Hill. I'm a research scientist at the California Academy of Sciences. And today I'm going to talk about some pretty preliminary work we've been working on for developing dynamic sampling prioritization maps for community science campaigns using species distribution model uncertainty. So community science campaigns or citizen science, community science produce a lot of data. And as we know, a lot of these data can be redundant for a lot of the questions that we're interested in. And when community science campaigns are developed, if they're not developed solely for engaging different communities, a lot of the time they're trying to fill different biodiversity gaps, geographic or taxonomic. A lot of these campaigns rely on expert knowledge to identify gaps, and this can be pretty difficult to do at very large scales. So, for example, our department at the California Academy of Sciences runs the City Nature Challenge alongside the Los Angeles Natural History Museum, and we've been thinking about how we can get people to collect more meaningful data than just more dandelions in San Francisco. But that's hard to coordinate across, this year we had more than 600 cities participating all over the world, and it's hard to coordinate particular sampling priorities across all these areas. So one of our big questions is, how do we develop a sampling prioritization tool that can scale to large geographic and taxonomic scales? And so we've been hearing a lot about adaptive sampling throughout the conference, and so I'll kind of skip over this, but the main takeaway is that yes, adaptive community scientists respond positively to adaptive sampling. Corey Callahan showed us that. Model uncertainty is an important metric in adaptive sampling methodologies, and while targeted questions and taxon specificity is probably the most important, we think that there's an opportunity, again, like for the City Nature Challenge, for kind of dynamic prioritization maps that work at large scales and across large geographic and taxonomic scales. We think there's an opportunity for that. And so I'll skip over this in the interest of time. So what you're looking at here is, I'm probably blocking a little bit of it, but what we're looking at here is a map of all California vascular plants that have more than 75% of their observations within the boundaries of California. We trained and bootstrapped maxent species distribution models for each of these species and took the standard deviation of those predictions and came up with our uncertainty maps. And the way you can kind of interpret this is these areas in darker blue are areas where if we ran bioblitzes there, collected additional samples there, we would have a better understanding of the niche and distribution of California vascular plants on average. But of course, when a modeler produces these prioritization maps across a ton of taxa and on a large scale, you can, a user or a community science campaign organizer can subset them to particular taxon of choice. And you can see that for oaks and Mariposa lilies, these prioritization maps look different across the state of California. And it's also trivial to subset them to particular geographies of interest. This is the San Francisco Bay Area, and you can see kind of relative sampling prioritizations just within this geography. And then you can also overlay accessibility layers, which are really important to community scientists. These are all the public lands in the San Francisco Bay Area. And someone that's interested in running a BioBlitz, or even users themselves, can look and say, hey, this is a parcel of public land where there's high uncertainty and that we might run a future BioBlitz in. And Rancho de Corral de Tierra Park, south of San Francisco, was one of those spots that looked really promising. And finally, again, this is very preliminary work on our end. We want better SDMs that incorporate more than just the 19 bioclimatic variables. We think a lot more might be important. We want to scale to more species and geographies. I mean, I just started with California plants because that's where my expertise is. More accessibility layers, thinking about roads, distance to roads, different transit options as well. And then eventually producing a dynamic web app, like something that Michael Pocock showed yesterday with the Decide web tool, but I think oriented more towards community science organizers and kind of at a larger geographic scale. All right, thank you very much.
Okay, last but not least, we have Gabrielle Dansereau discussing optimizing sampling and monitoring of species interactions within the biodiversity observation networks. Gab?
All right, so hi everyone, thank you for making it to this last presentation. Really happy to be here. And here in discussion, I want to contribute and bring consideration for species interactions to this interesting discussions we've been having about sampling and monitoring optimization. And so I'll be focusing on this question that you see over here of how we should optimize sampling to retrieve the interactions of a focal species. So first, why should we want to do that? Well, species interaction are a very important component of biodiversity, and also one that might be a bit more difficult to sample and document than just species ranges and species presence and absence. And so what I'll be showing here is a result of a simulation experiment that we did, in which we chose to focus on the case of a focal species because it gives us a clearer way to evaluate the efficiency of different monitoring strategies that we can have, while also being representative of an approach that we could actually take on the field. And so what we did is we generated a set of species ranges, including for our focal species. So think of it as a simulated landscape, such as the one that we would have over here, where our focal species would be present. We also generated a meta web of interactions to list all the possible interactions of our focal species. And we have also the ranges of interacting partners. So this means that in our landscape, we have spatial variation in both species and interaction composition across the landscape. And over this, we designed the different bonds, biodiversity observation networks, where we could go to sample and monitor the interactions of the species. And so I'll be comparing a few optimization layers, so ways that we could try to optimize the monitoring here in terms of the proportion of monitored interactions that we would have for this focal species as we increase the number of sites in our bond. And so the first case I want to highlight here is what if we use species richness as the element for optimization? So if we knew that the richness and all location in the landscapes, the total number of species, and we use that to design the bond. And so in terms of the proportion of monitored interaction, this was not the most efficient strategy in our case. It's possibly, and well, quite possibly, because with that, we're targeting location where the species is not present in this case. But it's not a trivial example, because it's useful to highlight what could be happening with a bigger program. And also it's useful to highlight that sometimes higher level and like community level information that are more required to, more difficult to acquire, are not necessarily the most informative for some specific monitoring goals. Since in contrast, in our case, using the focal species range, so the range of our focal species, was actually a more efficient strategy in this case, while also representing possibly a more attainable level of information. Given with SDMs, we can have a pretty good idea of the distribution of many species, and so this can be a relevant target for optimization. Finally, the last case I want to highlight, the one that consistently performed the best in the simulations that we try, was when we use as optimization layer, a layer containing the realized interactions, the location of every realized interaction for our species across the landscape, which is really the one that performed by far the best in terms of quickly reaching a high proportion of monitored interactions and also a higher proportion in general. And so to focus a bit, this is zooming on this last panel that I had over there, what it looks like of where the realized interactions are across the landscape. The question we can have in this case is, well, is this really an attainable level of information? Quite probably not, but anything that gets us closer to this could be useful in this case, and it's informative because it tells us that maybe the factors that are influencing interaction realization should really be the one that we try to prioritize as we try to optimize our monitoring. This could mean, for instance, better comparing species interactions, species abundances, sorry, into our models. And so my two take-homes for this presentation here were that, well, sometimes attainable species-level information, such as species ranges, can perform better than community-level ones. But finally, that additional efficiency gains will really require better knowledge of where interactions are likely to take place and incorporation of those into our models and optimization attempts. But thank you. I'm pretty sure I don't have time for questions as anyone, but I'm happy to chat about it.
So I think what we'll do is have kind of a discussion question session in a minute. But first, I just want to thank all of the speakers from this afternoon, from this morning. It was a really good session. I'm really excited. I have a lot of ideas just like percolating. I love that we talked about species, we talked about traits, we talked about interactions, and the idea that we can like optimize for all of these things at once is really exciting. I also want to thank my co-organizers, so Tanya Berger-Wolf sitting here, David Rolnick, who's there in the back, and Catherine Hebert, especially Catherine. She really did everything. So we have until 3 for discussion, but I also want to give some of you the chance to leave if you want to, if you don't want to sit through this discussion. And everyone else, I want you to come like closer. You don't have to come too much closer. So I'm just going to leave a few minutes. So decide what you want to do. If you're done, just leave or come a little bit closer. If you're a speaker, you have to stay. Okay, is everyone reassorted? It's kind of like church. Everyone's still in the back. So let's just start with any questions we have for the last set of speakers, and then I'm going to open it up. I have some broader questions that I want to ask the audience. But does anyone have a question for the speakers in the last session? So Gab, Michael, Millie, other people? I teach undergrads. I'll wait. No, no questions? Okay. Oh. All right, Catherine's going to ask some hard questions then.
Hey, everybody. Whoa, hey. This is different from karaoke. Oh, my God. I have kind of a Kickstarter question that's an open question. I really liked this session because it was a very solutions-oriented session. It was very optimistic. A lot of people are thinking creatively about how to fill the data gaps that are bothering all of us. And we saw solutions that were user-oriented, like figuring out ways to give people more information about what is helpful for science. We saw ways of incorporating that into our models and our frameworks. We saw other stuff too. A lot of technologies to link information to users to help them make better observations for science. I'm wondering if you guys are more inspired by some of these solutions. Do you see more connections that we're not making? Are you particularly excited about some of those connections or applications that we saw? Like the iNaturalist pilot is very cool to me, I find. Very localized information about what's important. Do you guys think that's part of the solution? Woo! Oh. Are there solutions that you didn't see here that you're like, why is nobody doing this yet?
Hey, we are going to get you talking. So first of all, just to, even from my visual mapping, can the speakers at least raise their hands so we know where they're concentrated? Okay, here's the sampling. Okay, great. So you are required to at least try to answer the questions. Everybody else, please ask questions. But the questions I want to kind of segue from Catherine's, there was a lot that was presented, and as sitting and listening to the talks, I'm like, oh yeah, this is the sampling strategies that we should be using. Yay, wait, no, this is the one. Oh, but if we want to answer that question, that's the one. So I feel like I need a little bit of a flow chart. What's my question? How do I get to, which are the strategies or the things that I should be thinking about? How do I choose? What should I ask? What are the things that I should consider, sort of things? Raise your hand if you felt the same way. I don't know. No, a little bit. So the question to the speakers, how would you think about connecting or choosing the sampling strategies in context of, and how would it change in different contexts of making decisions, different kinds of decisions? Who wants to start?
Okay, I'll provide a bad answer so people can provide better ones, maybe. I don't know. I feel like a lot of the talks at this conference were cool because they highlight some of the endpoints of the data, right? Like the EBVs and the framing things in terms of global policy and such. But I feel like probably the best approach is starting from there and designing things backwards with a specific set in mind. So I know in Canada, the stuff that we think about, everything or most of the stuff we do in Laura's lab, I think, is framed under the pretext of building better species distribution models a lot of the time, which obviously helps with area-based planning and stuff. But I think I think probably approaching it end point, then reverse engineering all the way back to the adaptive sampling or whatever sampling strategy is probably the right way to go. But yeah, that might not be true for everything. I'm not sure.
Yay, we succeeded. Millie, you raised the hand, right?
Something. Yeah, I mean, I couldn't agree more. I think the reverse engineering thing is really, really critical. And I think yeah, there's an interesting question of if we reverse engineer from the decision or from the ecology or somewhere in between, because always the data obviously isn't directly feeding into the actual decisions we're making. So I don't know, this is something I'm grappling with a lot at the moment. How useful is it actually to back engineer from the decision? I had something else to say, but I can't remember what it was, so it's Friday afternoon. And,
I'll run the microphone back to you.
I just wanted, on that note, to maybe give a little bit of context, which I had in an intro part that I didn't actually end up on the itinerary. But I think what you said is really important because we have different goals, even with adaptive sampling. And like adaptive sampling itself has been around for a long time. And it kind of started in adaptive management. And the point there was like, hey, we're not collecting data to answer the question. And that like has been talked through, and then they did all the stats on it. But it only ever manifested in some really good, but only a few case studies. And then there was like this general kind of what's known as surveillance monitoring, which came along a bit later. And it's like, oh, we want to know about everything. And that's kind of the adaptive monitoring part of that. But that's harder because you don't have really specific goals. So then it's hard to reverse engineer. So then there was this whole debate between like, is it, you know, should we do surveillance or should we do really targeted, more management things? And I think now, like, we can kind of do both in a way with like the technology, but we haven't really, in my mind at least, thought through how that's done because there's a structure to that. And people, you know, like Corey have written amazing like conceptual papers on how to do this, but I feel like with the people in this room, we could like devise something that's like a structured way to like, we want to do the surveillance, we want to do the targeted monitoring when we need it, and we want to like take into account that like these are communities doing this. You know, and I feel like that, putting those pieces together would be really cool. I mean, oh, yeah, and then go back to it.
And to add to it, now we're using machine learning AI models on top of it to derive it. And to channel Sarah Beery, if you want, there is the layer of, okay, what should we sample, what data should we bring in to change the outcomes of these models or improve the performance of the models. So there is a whole different dimension of consideration of what you sample and where. Because if you sample and the model outcome, and use the model to predict something and the model outcome doesn't change with any additional data, then you're just getting more data on the things that the model is already pretty confident about. So that's a consideration. I saw your hand.
Hi, I'm Matthew, and I come from a world of monitoring outcomes of conservation projects. So folks doing some kind of action and then doing long-term monitoring related to what happens. And I've been trying to figure out how it is that those data fit in to your world. And like how much value do they add? Because at some level it's data from the same site through time. So at some level you're like, oh, we already got this one, it's covered. Like, but I am really interested in the long-term species distribution model and the trends in, that connection is something I'm really interested in, and it involves all of these frontline communities, small NGOs, the like tips of your government agencies that are out there working on a marsh, and they're all collecting data. So I'd love to figure out how to incentivize them to offer their data for your for this global effort.
I mean, first of all, just to say, like, we have all this deluge of data we're collecting kind of everything for, but those targeted things are invaluable. Like, there's so much we can learn because we still can't, and someone maybe will disagree with me here, but if you take citizen science data, almost always, you can't detect a trend. It's nearly impossible, possibly in the future, or maybe for specific examples, if something's moving quickly, a range shift, you can detect it. Other than that, you can't. You have to have this community-based monitoring. And so I think it's hugely valuable, and it's not a piecemeal, and it's also valuable to the community, right? It gets people engaged.
But they're not, the people we're working with aren't thinking about y'all at some level. So it's just really interesting, and I think that I'd be really interested in trying to find ways to incentivize the people we work with to share their data, because at the moment they're not. They're using it for their own purposes, right? And so we are trying to figure out how it is that we can make it so that we can pipe data to the places that you guys are pulling from. But I don't know, I'd love to figure out how it is that we can help incentivize these folks to do that work.
So I'm going to repeat what my call to action from yesterday, and channeling also Sabine Leonelli's keynote on Wednesday, that a lot of these structures and infrastructures, we've put around biodiversity data and the protocols for monitoring are very one-directional, from communities to kind of increasingly more and more aggregated metrics. Even the aggregators like GBIF, you know, it is, or a naturalist, it is very one-directional pooling. There is very little flow in the other way. And one of the incentives in my world of AI for biodiversity, there is no AI without data, repeat after me. But if we're pulling that data and training some AI models, then there is no backflow from the insight and sort of expanded capacity that the models give, because quite often they're not just the advantage of the modern AI models is that they're not just this super coarse resolution. They actually can get pretty both increase the scale while maintaining the super high resolution across time, space, and individual organisms. So how do we build infrastructure that propagates that value back to the very, very local communities, especially if they're not resourced for AI, but are resourced for data?
Just want to say like a nuance, because there is this, you know, the information that goes in only one direction, and then it goes from the communities to ourselves here, scientists and so on. But there is a nuance here, which is the parachuting that we all do a lot. So it's not the communities that's collecting the data, it's ourselves going to one place, measure everything we can measure, and we go away and we work with people locally, but then we come back to our countries and we never recontact these people anymore. So there is also this parachuting problem that, at least in my community, is really critical. And we need to try, you know, fix it, working with the communities.
All right. All right.
Hello. So switching gears just a little bit, I'm kind of curious. So, you know, data constrained scenarios, we only have limited resources. A lot of the talks have been around adaptive sampling. How can we maximize the utility of our sampling? But that's sort of based around the utility of a given observation or a given piece of data we're collecting. And so part of these solutions hinge on like the informativeness, you know, the modular, I forget there was a definition someone had in the earlier session. And so I'm kind of curious, do we think that there's a universal way that we can measure the informativeness of this data? Is it only going to be for a given decision or a given management task? Or is it somewhere along this gradient?
Yeah, well, I think that's really interesting. One thing I kind of wanted to mention in my talk but couldn't is that, and I think we saw this reflected across the different talks, is that the different types of sampling were siloed into like either geographic sampling or sampling for specific taxa. And I think something that would be very interesting is like, given the way that a lot of this data is collected through community science, given someone is in a given location, what species would be most informative for the model? So like there is multiple ways to define that, so I think that's really useful, but finding a way to sort of combine each of these different ways that we're structuring what counts as optimal sampling, I think would be a good improvement for sort of having a more general theory that sort of incorporates all these different things.
And to add to Matthew's and the previous comment, if you're bringing AI into the conversation, then you cannot actually define a static value of any given data point. It has different value for at different time points depending what you've already put in and where in the timeline of prediction process you are.
Yeah. And just a more maybe general answer to that question, we still don't know what we don't know. I mean, Target 21, it is in the Global Biodiversity Framework, and we're kind of struggling how to measure it. So I think there are very systematic ways we can do it, but it just hasn't been developed. So far, it's just like really basic indices of gaps. That's what we've really done so far. So I think it doesn't have to be overly complicated, but it's probably not going to be like one set of metrics. But okay, with that, we're at the...
I have one more thing. Sorry. Hi, I'm Rebecca. I've been thinking there's so many amazing things that everyone talked about today. And in my group at the California Academy of Sciences in San Francisco, we do all the parts of what we've been talking about. So we run campaigns to get people to collect data, then we analyze the data, and then we bring those data to decision makers. And so my mind is like completely racing. But I think there are a couple of really important things. One is that there's a question about how observation and individual observation might matter for a model. But we're all working with data that individual people collected. And so I think it's really important to think about how each observation matters to that person. Like, even if it doesn't improve our models, like we're also trying to build a community of naturalists that care about this work. And when we think about how to make that, to bring data back to people, it maybe isn't just like, thanks for improving our model. Like, I think there are other ways that we can communicate the value of what we're all doing with those data to help people answer questions they might have. People are using iNaturalist data right now at city council meetings to argue for X, Y, or Z, or like they're being used. And so if we can provide the tools that would help at that local level, even templates to do that, that would be amazing. And then I think one last thing, for decision makers, I think there's so many different levels, and I love how Millie's thinking about this. Like, we can start with questions that decision makers have, but a lot of times, as you maybe all know, they're making decisions based on like one story or like expert opinion. And so how can we recognize how they're actually making decisions? And a lot of times they don't know the decision they need to make until they have to make it. And a lot of times the things we're doing are not at the scale that they need to make decisions. And so all of these things I think we can think about in this kind of like framework that's kind of being discussed a little bit.
That's a perfect place to end the session. Thanks, Rebecca. Thanks, everyone, for joining us and sticking with us to the very end of the conference. So enjoy the coffee. See you later for the happy hour if you want to come. Thanks again.